My name is Harriet Alexander. I'm an assistant scientist at Woods Hole Oceanographic Institution, and my lab focuses on computational approaches to study microbial life in the oceans. Today, I'm really excited to be giving a talk for the BVCN focused on a program that we've been developing, and the title of my talk is a Eukaryotic Heist, Scalable and Automated Approaches for the Discovery of Eukaryotic Genomes. So I'm an oceanographer, and the organisms that interest me most are the microbial organisms that live in the ocean. And one fundamental problem that we have in oceanography is that we have these complex mixed communities of these organisms that are fundamental to the functioning of the ocean. And yet disentangling what individual organisms do is a really complicated process. However, with the advent of different types of metaomic approaches, such as metagenomics, metatranscriptomics, we have begun to be able to shed light on these complex mixed communities. However, ultimately, it's computational biology, or the process of the analysis of these data, that actually allows us to begin to get at these fundamental questions in biological oceanography, such as what are the ecological traits of these organisms, what is their general biogeography, and how do they influence ecosystem processes? And broadly speaking, computational tools and approaches for the study of protists or eukaryotes really have lagged relative to those for bacteria. And I would argue that this is especially true for environmental metaomic analyses. So why care about eukaryotes? Well, eukaryotes in the ocean are diverse, and they're hugely important to our understanding of marine ecosystems and biogeochemistry. And this is a figure that I love from Alex Warden, which shows the variety of different behaviors or functions that these organisms can play in marine systems, ranging from photoautotrophs, tiny plants, all the way to heterotrophic organisms that graze on bacteria and other organisms. Broadly speaking, my two main interests are what are these biological responses like to fine scale oceanographic features, and what is the ecological role and the prevalence of eukaryotic pangenomes in the ocean? However, I would argue, and since this is a slightly broader audience than I usually give talks for, that eukaryotes are diverse and hugely important in other ecosystems. And this is pretty well defined in host and terrestrial ecosystems. Here I'm showing a wonderful figure from Parfee et al. 2011. And this is just a phylogeny of different eukaryotic taxa, and they've highlighted groups which are present in human gut or invertebrate gut or human parasites in other regions across this tree. And you can see that these organisms are diverse and they span the eukaryotic tree of life. And here are some micrographs from different gut-associated protists. And within the human literature, it seems like there's an increasing tendency to call this the study of the eukaryome as opposed to the microbiome. I'm not sure if we should adopt that in oceanography or not, but I kind of love the term. And so the eukaryome is really important, but it's broadly less well studied than its prokaryotic counterpart. Partially because it's hard to study, and also because these organisms are really challenging to culture. And this is true not only in the oceanographic sense, but particularly from terrestrial systems or microbiome or host-associated systems. And largely, especially within human-associated systems, we have a better understanding of the parasitic organisms and less good understanding of those that are commensal or potentially beneficial to us. And while there are a lot of metaomic approaches, a fundamental problem that we have in the study of eukaryotes within these environmental systems is our lack of genomic references, which really complicates the interpretation of metaomic data. And here I'm showing a figure from Keeling et al. Related to the advent of the Marine Microbial Transcriptome Sequencing Project, or MMETSP. And this highlights the different branches, the solid branches where we have a sequenced genome, and the red dots represent branches where we have sequenced transcriptomes. And so we do have a lot of branches on the eukaryotic tree of life, which we've identified, yet we don't have genomic information on. We don't know what genes they possess or what combination of genes they possess. And so one question I have, and a question that kind of motivates this entire talk, is can we begin to leverage these environmental omic data sets to begin to improve eukaryotic reference databases and ultimately our interpretation of metaomic data? So a brief overview of my talk today. I'm going to begin by explaining the EukHeist pipeline development. I will then go into an example of an application of this particular pipeline for the recovery of MAGs, or metagenome assembled genomes, from Tara Ocean's large size fraction metagenomic dataset. And then I'm going to provide one fun vignette, which is a little ecological insight into the role that eukaryotic MAGs are playing in the ocean. Eukaryotic organisms. The recovery of MAGs, or metagenome assembled genomes, is becoming an increasingly popular and broadly applied approach across the field of microbial ecology and microbial bioinformatics. The general idea is that you start out with a set of metagenomes from an environmental sample, and you sequence your genomes, and ultimately you combine samples or not and assemble those samples. Then there's a statistical approach whereby you assess both the tetranucleotide frequencies, which tend to be unique to different lineages of organisms, as well as the relative abundance, to back calculate the likely genomic content, or complete genomic bins, aka MAGs, which were present in the original samples. So the data set that we're working with and talking about today is from Tara Oceans. Those of you in the oceanographic community are undoubtedly aware of this Herculean effort. This was a global sampling effort that went around the world's oceans on this sailing vessel pictured here, following the track that's pictured here. And they did global consistent sampling of the ocean's plankton, meaning that at every station they sampled the same types of things in very consistent and reproducible ways. And these data have been used to great success to start to illuminate a lot of the functioning of the microbes and the plankton within the world's oceans. And in particular, I want to highlight two different studies which did large-scale recovery of MAGs, or metagenome assembled genomes, to shed further biological insight on the functioning of these organisms. The first, by Delmont et al., looked at these data to identify this kind of novel niche of nitrogen-fixing heterotrophs. And then Tully et al. Did this wonderful work looking and recovering over 2,000 different MAGs from these data. And so the question is, can we begin to actually optimize the binning process to target eukaryotic genomes? And there was a fantastic paper published in 2018 by Eukaryotic et al. Entitled A Global Atlas of Eukaryotic Genes. And this represented the data from the large size fraction from Tara Oceans. And this was, again, uniform sampling of both RNA, or metatranscriptomes, and DNA, or metagenomes, from across the ocean. They sampled from size ranges from 0.8 microns all the way up to 2 millimeters at the surface, deep chlorophyll maximum, and mesopelagic, so different depth layers within the ocean. And then from these data, they assembled the metatranscriptome and characterized over 116 million unigenes, or novel genomic units, from the ocean. And this represents the largest collection of eukaryotic transcripts and gene families recorded to date. It's really a fantastic advance for our field and our understanding of eukaryotic biology. However, the initial analysis really focused on that set of metatranscriptome assemblies and didn't dig too deeply into the metagenomic data. And there are a couple of reasons for this. Larger size fractions are really dominated by eukaryotes, so plankton, small animals such as copepods, etc. And eukaryotic genomes are far larger and much messier than prokaryotic genomes. Eukaryotic genomes are dominated by introns, tons of repeats, pseudogenes, etc., which really complicates the assembly and analysis of these data. However, by studying the poly-A selected mRNA, or the transcriptome from these organisms, we're able to focus specifically on the genes themselves and not worry about all the messy in between. However, these data are really exciting. And so what we wanted to do was go in and actually start to look at the metagenomic data. And the motivation was ultimately trying to harness this environmental data to improve our interpretation of those data. And so our motivation was basically twofold. First, there was the biological, we wanted to expand the catalog of eukaryotic genomes independent of cultivation to get an idea of functional traits, but also to begin to look at and try to understand potentially these non-coding regions of the genomes. There was also the technical. We wanted to see if we could create an automated, scalable, and reproducible pipeline to study these metagenomes and start to recover these eukaryotic potential MAGs. And again, as I said at the beginning, I would argue that eukaryotic focused pipelines are really lacking within our field. So since this is the BVCN, I am doing a BVCN special aside on reproducibility. So I would argue that there are kind of two main practical problems in computational biology or computational science, regardless of your specific subfield. First is repeatability. Let's say you do an analysis and then you go away for a couple of weeks and you come back, or maybe you go away for a couple of years and you come back. Can you do it again? Can you do it again on your computer? Can you find your scripts to accomplish and recreate the figures that you accomplished or created initially? And then there's the issue of reproducibility. Can someone else do it at all? Can they find your scripts? Will they be able to get the same answer? Will they be able to understand and reproduce your results? And this is really important as we start shifting towards these more computational approaches to understanding biology and ecological systems. So let's take a little trip down memory lane. This is a paper that I published back in 2015. The title was Functional Group Specific Traits Drive Phytoplankton Dynamics in the Oligotrophic Ocean. And here's a figure from the paper. I'm not going to talk about the results of the paper, but rather the process. So I started with 24 meta-transcriptomic samples that I wanted to process in the same way: QC, align, assemble, etc. And so I asked the question, how did I do this back in 2014 when I was in graduate school? Well, this was one of my first computational projects. And I would say that I took what I would like to call the Mad Libs style approach to bioinformatics. So I created templates for various scripts that I wanted to execute on my local HPC. I left a blank or kind of fill in the blank for the sample names. And then I auto-generated scripts to basically do the same thing to all the different samples and ultimately hope that they don't fail. So what did this actually look like? Oh, and of course I had to rinse repeat for every single step of the pipeline. So every step that I needed to do, I had to generate all these new scripts for each sample and execute them. So this is an example of what this looked like. This is a shell script that I wrote to do some kind of parsing and finding and renaming of files for the generation of a given script. So this is my Madlib scripter. And this generated all these different scripts. You can see these are an example of some BWA mem.sh type scripts that could be executed or sent to whatever HPC I was using. So how did 2014 Harriet do? Well, the good. I conserved sample names, which is nice. The scripts were not completely manually generated, which means that there's less chance for human error, which is a pretty good thing. And I know where my analysis scripts are, which is also a plus. You got to know where your original analysis scripts are. All right, the bad. I have no version control. 2014 Harriet was not on GitHub, didn't know what GitHub was, and definitely wasn't really in the open science community. I never defined my compute environment. I wasn't using any sort of Anaconda-based environment control mechanism. And these scripts were absolutely designed to work on one and only HPC. That was the HPC that I was working on at the time, which no longer exists. So that's problematic. And I would challenge anyone to try to run these on another HPC without having to change stuff, because I'm sure you will have to. And the ugly. All right, these scripts were ugly. They were really hard to interpret. They were hard to read. I didn't write a README file to help the poor soul who wants to actually run these down the line. So I give it a 75% chance of repeatability. I think I could get back into my head, move stuff around to make it work for what I'm using currently on my new HPC. And I give it a 10% chance of reproducibility. I would commend anybody who can actually carry that off. So overall, I give this analysis a C+. All right, so why do I say this? What if I had more than 100 samples? What if I had 1,000 samples or 1,000,000 samples? It just would not scale. This process is being driven by a human brain, and my brain is not capable of checking and carrying out analysis on 1,000,000 things. So enter workflow managers. Workflow managers. What are workflow managers? Workflow managers manage and track or execute multi-step operations that are complex. And I argue that they really do enhance portability across systems. So a couple examples just from the literature that are being commonly used in the bioinformatics sphere. You have Nextflow, you've got CWL, you've got WDL. These are all great workflow managers. And I recommend checking out Writer et al. 2021. They've got a great review on workflow managers in general and go into more depth than I'm going to. But today I'm going to be talking and sharing with you our pipeline, which was built on SnakeMake. So SnakeMake, why? Well, you've got a lot of choices when it comes to workflow managers, but the reason I like SnakeMake is that it's lightweight. It's very flexible. It's written in Python, which is a language that I'm very comfortable in. And so I found it less intimidating to start kind of getting into that particular set of syntax. And there's a huge, large community of users, meaning that there's an active response to questions. There are people posting stuff on Stack Overflow. It's just a great community that's very responsive. And I saw on their website that they're getting more than 5 citations per week of SnakeMake, which is really impressive and speaks to the community involvement. What does it look like? Well, essentially it looks like a Python script. You can use straight up Python in various components of SnakeMake, but the SnakeMake specific portion are basically these rule definitions. And here I'm showing just a random rule example called count words. And so this takes some input. In this example, it's siddhartha.text. And then it will look for some output, say siddhartha.dat. And then in terms of what it's supposed to do, I have this little shell command written down here, which is word count, give me all the lines in our input, siddhartha.text, and save it to the output. And so the nice thing is that SnakeMake basically is able to fill in the blanks for you. So as compared to my Madlib example, SnakeMake does that automatically, and it's kind of hidden, and you're left with this very clean looking input-output structure. This is a very simplistic example of what SnakeMake looks like, but if you want to check out more, they have a wonderful Read the Docs website. Back to the actual project at hand, which is the design and implementation of the EukHeist pipeline, which is the scalable and automatic retrieval of eukaryotic metagenome assembled genomes, or MAGs. So at the basic level, the EukHeist pipeline, which is written in SnakeMake, is much like any sort of metagenomic flow pipeline that you might see out there. So starting with Illumina sequence data, which are short reads, 100 base pair, paired end. We do basic QC as well as trimming, and then move through various steps. And I would say that when you're considering a metagenome assembly plus binning project, the 2 major steps that you need to do a little bit of consideration of are the short read assembly and the contig binning. There are a lot of different tools that are out there which take different approaches and have different drawbacks that you can choose for both of these 2 steps. And so this is where I would say that metagenomic pipelines tend to vary quite a bit. And when I was beginning the project, I spent a lot of time looking at the literature, looking at various different metagenomic assemblers. And around the time I was starting the project, the CAMI project, the Critical Assessment of Metagenomic Interpretation, had just been published. And if you haven't looked at it, I highly recommend checking out this paper or this project website, because it's a continuing endeavor, continuing to provide assessment of different metagenomic tools. And here I'm showing different outputs that they had from the project. First, for comparison of different assemblers. So just looking at the genome fraction that were properly assembled for different genomes within the data sets, as well as the performance, the recall versus precision of different metagenomic binners, with the gold standard being up here in the corner. Another resource that I really enjoyed while I was starting out on this project were the blogs that have been put out by the Marin Lab, Marin Lab being the group that developed Anvio. And here I'm just showing two wonderful figures that they generated, one comparing the fate of different contigs as a function of different the different binners, and the other showing the relative distribution of reads based on different read recruitment algorithms or read aligners. So I highly recommend checking out his blogs. They're really informative and fun to read and very thoughtful. Anyway, back to the question at hand, which for this particular project, I knew that I wanted to do large co-assemblies. And so for me, memory usage was a very big consideration for the assembler. And so I ended up deciding to go with MegaHit because it's really good. It's very fast, relatively speaking, and tends to use less memory relative to other large assemblers or metagenomic assemblers that are available. And then for the binning of contigs, at the time I chose MetaBat2 because it seemed like a very robust option. However, now with the advent of things like DAS tools, there are a lot of different binning options, which are really fantastic. And so if you're thinking about starting out on this type of project, I really encourage you to check out the literature and see what people are doing currently, and also check out some of those blogs that I mentioned and the CAMI project. Anyway, so SnakeMake basically drove this entire analysis pipeline, and what this actually looked like for the Tara project was this. So this is a summary of all the jobs that were submitted to my local HPC based on SnakeMake. So we had 94 BWA indexing, we did 94 MegaHit assembly jobs, et cetera. And you can see that in total, it ended up being 6,000 jobs, which were submitted just for the basic run of this pipeline, which really is better than me doing it by myself. And this pipeline is currently tested with Tara. We're working on other data sets, but it is designed to really accommodate any sort of paired meta-G, meta-T type data set. So taking a look at the data, this is a min-hash comparison of those 824 Tara Ocean's large size fraction metagenomes. And here I'm showing a plot which shows the Jacquard similarity between each of those raw sequence data files. This is a tool from the lab that I did my postdoc in. It's called SourMash. It was developed by Louise Erber and Titus Brown. And what I'm showing here is where it's bright, it means two samples are very similar. Dark means they're more different. And I'm coloring the samples based on the ocean region that they came from. And so at first glance, you don't see any major coalescence among ocean regions. However, if we layer on depth and size fraction, you can begin to see that broadly, samples tend to group based on their size fraction and their depth. And so we decided to ultimately co-assemble our metagenome samples from the same region, depth, and size fraction, which took 824 samples encompassing more than 20 terabytes of raw data into 94 distinct metagenomes. And as we were starting this process, we figured that there were three possible outcomes. First, the pessimistic. Nothing will assemble or bin well because we're in eukaryotic dominated samples and it was complete time wasted. We then had the likely, which is that we would recover mags from particle or plankton associated bacteria and archaea because they have smaller genomes. It's been done before. It seems likely that we're going to get those out. And then we have the hopeful, which is that we would recover mags from marine protists and small metazoa and really get at those eukaryotic organisms. So from these 94 metagenome assemblies, we recovered 16,000 candidate bins. So how well did we do? What did these bins look like? Here I'm showing a scatter plot of two different single copy gene metrics. On the x-axis, we have check-M completeness, which is a standard completeness check, which is used for bacterial and archaeal mags. And on the y, I've got the Busco eukaryotic gene set for completeness. And what you can see is that there's this population of bins which are rising above the fray and appear to have high eukaryotic completeness. And if we color this by mag size, what we see is that not only do they have higher eukaryotic completeness, but they also tended to be much larger, which is an observation that has been made previously by Delmont in 2017. And so it seemed likely that we were, in fact, recovering some putative eukaryotic mags. However, we wanted to do this more systematically. So what we ended up doing was devising this flow chart of eukaryotic versus prokaryotic mag differentiation. So starting at the top, the first question we asked was, is the mag larger than 2.5 megabase pairs in size, which is roughly the size of the smallest known eukaryotic genome? So from our 16,385 bins, 4,777 of them were larger than that cutoff. From there, we used a tool developed by Patrick West called EukRep, which estimates whether a contig is likely eukaryotic or prokaryotic in origin. And so we required that over 90% of the content in a given bin be estimated to be eukaryotic. With that cutoff, we ended up with 988 putative bins, which were both larger than 2.5 megabase pairs and had high eukaryotic content. From there, we then looked to our complete single copy eukaryotic gene sets via BUSCO completeness as well as EukCC. And we found that ultimately we had 485 high completion eukaryotic bins, which had more than 30% coverage of single copy orthologous groups for euks, and we had 503, which were low completion. And on the prokaryotic side, we recovered 4,021 likely bacterial and archaeal bins. And we are calling these mags, or this mag set, Topaz for the Tara Oceans particle-associated mags with a Z. So first off, just to look at the prokaryotic side of things, this is work that I've done in collaboration with Maria Pakiadaki and Ben Tully. And here I'm showing just a quick phylogeny based on concatenated genes of our high quality non-redundant eukaryotic mags. So from those 4,021 eukaryotic mags, 1,273 were high quality, meaning that they had greater than 90% completeness as estimated by CheckM and were less than 5% contaminated. And of those, 716 of them clustered at 99% ANI similarity, meaning we recovered 716 high quality distinct mags from prokaryotes. And it really spanned a huge diversity of organisms. And one kind of interesting thing we wanted to look at was, what are the genomic traits, or do these genomic traits differ between these likely particle-associated bacteria and those that are more likely to be free-living? And we found that compared to a set of Gorg SAGs, which are from Pakiadaki et al. 2019, the Topaz mags tended to have larger genomes overall and higher higher GC content. We also saw that there was overall enrichment of peptidases as well as CASI proteins, or proteins for the breakdown of carbohydrates, which is interesting and indicates that it's likely that these bacteria are able to capitalize on a different lifestyle than free-living pelagic bacteria. But for the rest of the talk, I'm going to be focusing on the set of high completion eukaryotic bins. And overall, the basic analysis of eukaryotic mags has really not been well established. We don't really have a good handle on how to do gene prediction. Taxonomy, classification of these bins is not straightforward. And also function is really not very well developed. And so a question we had to grapple with was, when and why do we need to start thinking about finding or building a new method? And I'm going to be talking about two different examples today. The first being gene prediction in eukaryotic mags. So identifying the coding regions for these eukaryotic mags, and the second being the taxonomic classification of these eukaryotic mags. And the question at hand was really, can we sort of retrofit existing technologies or approaches, such as those that are used for the study of prokaryotic mags, or do we need to go out and try to figure out a new method or develop a new tool to be able to do this well? And in the case of both of these, we ended up going with the new tool or approach, which led to new collaborations and new insights. And I think it's really been an exciting process. So the first tool that I'm going to talk about is Eukaryotic, which is a tool that's been developed by Chris Neely and Ben Tully at USC. And Eukaryotic is designed to predict genes accurately in eukaryotic mags. And this is hugely important because the analysis and interpretation of the biology of these organisms is really limited by the quality of our gene prediction. If we can't predict the genes in these organisms, we have no hope of really using these new eukaryotic mags to any sort of benefit. And on its own, eukaryotic protein prediction from genomes is challenging and imperfect. And here I'm showing a schematic from Eukaryotic, which is currently in prep for publication. But the basic idea is that you provide a set of input genomic contigs from your bin or your mag. It goes through and does an initial evidence-based search against a set of eukaryotic protein databases. It then calls an initial set of taxonomy using mmseq2 and uses that to inform the identification of some repeat maskers to do a good job of excising repetitive content from the genomes. And then it runs through a series of different ab initio prediction steps, including Augustus and GeneMark, to ultimately define a core set of likely protein coding regions. And so what this looks like is the following. So here I'm showing some benchmarking, kind of similar to what the CAMI project did. Down here in the pink, I've got the gold standard, which are NCBI eukaryotic genomes, specifically targeting non-plant and non-animal genomes from NCBI. And then I'm showing here, we've got BUSCO completeness, and then the change in BUSCO completeness compared to the reference, so the gold standard, and then the total number of proteins that were predicted. And I'm showing results from Augustus, Eukaryotic GeneMark prediction, as well as MetaEuc. And what we see is that Eukaryotic does a really good job of recovering BUSCO proteins that are present in the gold standard overall and has relatively few losses. I feel like that's exemplified by this plot right here. Also, Eukaryotic is covering a similar number of proteins to the gold standard gene set. However, beyond gene prediction, Eukaryotic really is designed to streamline eukaryotic gene prediction here in this run function, as well as refinement, meaning you can go back and reconsider, add more information to try to improve or enhance the gene calling in a given set of mags. And finally, the annotation. It will automatically run BUSCO as well as any set of functional annotations that you want to do all within the same ecosystem. So I really recommend checking it out. And I think it would actually work quite well, even beyond mags, for general eukaryotic genome annotation. So a second fundamental issue is that taxonomy of eukaryotic mags is really hard. In particular, most eukaryotic taxonomic surveys tend to leverage 18S or 23S genes. However, those do not assemble well. They were absent from nearly all of our mags. I believe we only recovered one copy of 18S. And broadly speaking, we have fewer references for genomes and transcriptomes than there are present for bacteria. So the sort of GTDB type approach really isn't viable in this particular case, or would require really heavy manual curation. So a student in my lab, Ariana Krinos, has developed a tool that we're calling Ukulele. It's a reproducible, modifiable eukaryotic taxonomic annotation approach, which looks at consensus style annotation at the level of protein. So you can provide it either with eukaryotic mags or eukaryotic metatranscriptomes. It'll go and do homology searching in protein space, do a best hit identification, identify the last common ancestors for a given protein, and then ultimately look across all those predicted proteins and give you a consensus annotation at some given cutoff value for your mag. And it's published in JOS and there is a Conda installable Python package available. So I encourage you to check it out if you're interested. So going back to our 485 high completion eukaryotic mags, we applied both Metasanity and Ukulele to these data set. And here I'm showing a maximum likelihood phylogeny based on 48 concatenated BUSCO proteins that were found commonly across our mags and the references. So this tree is colored by the protein consensus taxonomic annotation as provided by Ukulele. And just adding some labels to the tree, up at 12 o'clock, we've got our metazoa, we've got our fungi and amoebozoa around 2 o'clock, and diatoms are down at about 5 o'clock, and then chlorophyta are all of these different green branches. So first off, I'm adding color based on where a mag was recovered from based on size fraction. So I've got the smallest size fractions here in the light yellow and the largest size fractions in darker green. And what we see, which is really encouraging, is that we tend to recover mags from pico and nanoplankton, such as Micromonas, our little pico phytoplankton, from these smaller size fractions, whereas in the larger size fractions, we tend to recover metazoan mags. And broadly speaking, across our data set, the majority of eukaryotic mags were recovered from the photic zones, or areas of the ocean that get lots of light, the surface and the deep chlorophyll maximum. However, there was one region where we recovered mags from the mesopelagic, and these all tended to be fungal mags, which is quite interesting and requires some further investigation. And then if I layer on the final ring of where these mags were recovered from, this is ocean region, we see that broadly speaking, the phylogeny and distribution of topaz mags really meets expectations. We don't see any major trends in terms of ocean regions being biased towards one lineage or another. And just looking at some kind of interesting statistics by group. So here I'm plotting BUSCO completeness, and I'm doing box and whisker plots for each of the different higher level groups of organisms. And we see that BUSCO completeness really varied by taxonomic group, with fungi in particular and also chlorophyta tending to be more complete compared to other groups. And here I'm showing the total number of predicted proteins. And as we might have expected, our dinoflagellate mags tended to have the most predicted proteins, here with on the order of 20 to 30,000 on average. By contrast, chlorophyta down here in the light green tended to have the fewest. And these findings really are in line with what we know already of genome sizes in these organisms. And finally, I'm showing the percent of proteins that could be annotated with PFAM. And broadly speaking, some groups tended to perform better than others. In particular, chlorophyta and fungi both tended to have higher proportions of annotation than our metazoan mags or haptophyta and cryptophyta. And this may speak to some biases or dominance that exists in our current genomic databases and how we've defined our PFAM families. So finally, I'd like to share one ecological insight from eukaryotic mags, and that is estimating the trophic mode of uncultured mags. So I started with this figure at the beginning, but I'm coming back to it again because it really nicely shows the different styles of behavior that can exist within the ocean setting. Again, we can have these phototrophs whereby an organism gets all of its energy from light. You can also have kind of more heterotrophic organisms, predators, parasites, saprotrophs, where they get their energy purely from organic carbon sources. However, there's this really interesting group of organisms that exist within the ocean, and those are the mixotrophs, where they get their energy from some combination of these two pools. So we asked the question, can we predict the likely trophic mode of these mags based on gene content? So we ended up developing a random forest classifier that predicts trophic mode. And this was built on orthologous genes from KEGG, looking at their presence and absence. So we trained these data on a data set that I mentioned earlier, the Marine Microbial Eukaryotic Transcriptome Sequencing Project, which generated hundreds of transcriptomes across eukaryotic diversity. And we also got a couple more transcriptomes that are present within the EukProt dataset that's been put together by Daniel Richter. And we assigned an estimated trophic mode to each of these transcriptomes or organisms based on an estimate from the literature. And this was work that was really guided by Sarah Hu, who's a postdoc at Hui. And the model we found performed pretty well. So here I'm showing a confusion matrix from our model run. On the X, or along the bottom, we have the true classification. So this is the classification of a given transcriptome that we assigned it based on the literature, heterotroph, mixotroph, or phototroph. And then here along the Y, we've got our random forest classification based on our model. And we have, again, heterotroph, mixotroph, and phototroph. And so this diagonal here is basically our true positives. So a heterotrophic mag being predicted as a heterotroph, et cetera. And broadly speaking, the model performed very well for both heterotrophs and phototrophs. However, most of the error tended to be around the categorization of mixotrophy, where often mixotrophic mags were classified as either heterotrophs or phototrophs. And one question we kind of have still open is mixotrophy really a genomically classifiable trait? There are a lot of different methods or means that mixotrophy arises. And so it's an open question, and I think it's really interesting to talk about, and I'd be happy to talk about it with anybody who wants to talk about it. But to kind of complement this random forest classifier approach, we also created what we're calling a heterotrophy index, which basically provides a sliding scale trophy classification. So a very positive number indicates a very strong heterotroph, and a very negative number indicates a very strong phototroph, and you'd expect mixotrophs to be somewhere around 0. And so when we layer this on to our tree, what we see is that it does surprisingly well based on what we already know of certain lineages. So here in these outer two most rings, I've layered on our prediction of heterotrophy and phototrophy. So the random forest classifier is this inner ring, which is a solid color. And then our heterotrophy index is the outer ring in these kind of gradient of red and green, or pink and green colors. And this really illustrates the spectrum of nutritional strategies within each branch, and it adds a certain amount of information about the actual traits or functions that these mags might be playing in an ecosystem that would be absent otherwise. So I think it's a really exciting avenue. And broadly speaking, I would say that machine learning type approaches really have a lot of potential in terms of predicting functional traits of mags. I think it's not limited to this question of trophy, but probably could be applied more broadly in kind of interesting ways. So with that, I'd like to just leave you with a couple parting thoughts. First, global scale programs such as Tara Oceans, BioGeotraces are really increasing omic generation exponentially. And of course, as an oceanography, I'm most intimately aware of the large scale programs that are happening in the ocean sphere, but this is equally true of other systems. Because sequencing technology is becoming so affordable, we can really start to do these large scale programs that are going to generate invaluable data that we can begin to leverage in really interesting ways. And In particular, I think we can begin to leverage these data to really improve our reference databases. So many things are not culturable, and this is really a wonderful avenue to begin to improve that situation. And finally, I hope I've convinced you that the binning of eukaryotic mags is tractable. However, there's a lot of work that remains to continue to improve our annotation, our gene calling, and ultimately our interpretation of these data. And with that, I'd like to acknowledge my co-authors on this work. For the Topaz work, I've been collaborating a lot with some wonderful people, including Sarah Hu, Maria Pakiadaki, Laura Blum, Taylor Ryder, Ariana Krinos, Ben Tully, Chris Neely, and Taylor Ryder again, oops, as well as Natalie Cohen, who worked a lot on ukulele, particularly from the metatranscriptome side. And this work would not be possible without the freely open data that's been made by the Tara Oceans Consortium and the MMETSP. And with that, I can't take questions because this is on the internet, but I welcome feedback. I'm pretty active on Twitter, so please feel free to reach out to me at nectonforplankton or send me an email. And again, these are the tools and pipelines that I mentioned in this talk. And thank you very much.